Hi guys, I'm Aditi, and this is the Culture of Wellbeing's first lecture on an introduction to neuroscience. We're going to discuss a total of 9 topics, and which include what is neuroscience, second, early studies of neuroscience, the nervous system, fourth, parts of neuron, fifth, parts of the brain and their functions, sixth, neuron types, seventh, action potential and synapses, 8, neurotransmitters, and our last topic, research methods such as brain lesion and cellular, etc. Our first topic is it's going to discuss what neuroscience is. At the most fundamental level, neuroscience is the study of brain and the nervous system and their structures and functions. Our brain is a very complex organ and acts as a big computer. It is the root of human intelligence and is involved with processing and sending information from the senses in the body to the brain. Our brain is also what makes us human, as it allows us to store and create new memories and thoughts, and experience emotions. Human brain have 86 billion neurons that are involved in sending messages across the body to facilitate movement and thought. The field of neuroscience aims to 1. Study how these neurons connect with each other and the rest of the body. 2. Study how the nervous system develops. 3. Defects and disorders of the nervous system. 4. Combining different approaches and technologies to research treatments for diseases and disorders affecting the nervous system. 5. Study how cognition and human behavior. 6. The anatomy, biochemistry, molecular biology, and physiology of neurons and circuits. Neuroscience is an interdisciplinary field and lies closely with other fields such as medicine, mathematics, linguistics, engineering, computer science, chemistry, psychology, and even philosophy. There are many subfields of study inside the vast field of neuroscience. The main subfields include cognitive neuroscience, social neuroscience, clinical neuroscience, behavioral neuroscience, development neuroscience, affective neuroscience, computational neuroscience. We will, we will cover more about each of these fields in depth in our next lectures. The second topic we're going to talk about is early studies of neuroscience. First, we're going to touch on the ancient studies in neuroscience. Alcmene of Croton, an early Greek medical writer, dissected the eye and related the brain to vision. Alcmene also suggested that the brain, instead of the heart, was the organ that ruled the body, and that senses were dependent on the brain. In 4th century BC, Aristotle theorized that the heart was the root of intelligence and that the brain served as a cooling mechanism for the blood. However, as we know, the earlier studies of the brain were very unsubstantiated and not based on accurate research. During the Hellenistic period, Herophilus and Erasistrus engaged in studies dissecting the human brain, which further provided evidence for the primacy of the brain by identifying the cerebellum, ventrices, and the dura mater, which is a tough outer layer of tissue which covers and protects the brain. Moving on, the Middle Ages and the Renaissance. During the Middle Ages, Islamic medicine focused on mind-body interaction and brought new light on the need to understand mental health. Avicenna, a polymath who was regarded as the father of modern medicine, presented detailed knowledge about surgical treatments regarding skull fractures. His medical writing explained phenomena such as insomnia, mania, hallucinations, nightmares, dementia, and more. During the Renaissance period, Leonardo da Vinci drew life illustrations of the human skull. In the drawing to the left, the maxillary antrum can be seen, which is a cavity that Da Vinci was the first known artist to describe. Da Vinci also drew the first ever accurate drawing of the meningeal arteries that supply blood to the brain. In later years, he accurately drew the brain and cranial nerves. His drawings described olfactory nerves and cranial nerves, and illustrated the crossing of nerves at the optic chiasm. The third topic is about the nervous system. The nervous system is a complex network of nerves and cells to and from the brain and spinal cord. It is the main controlling, regulatory, and communicating system in the body. The nervous system is made up of two parts, the central nervous system and the peripheral nervous system. The CNS is made of the brain and spinal cord, and the PNS is made of nerves that branch off from the spinal cord, extending throughout the body. The nervous system's activity controls our body's ability to move, breathe, think, experience senses, and more. The basic unit of the nervous system is a nerve cell called the neuron. Neurons can perform different activities, like transmit messages from the brain to the muscles to facilitate movement, detect senses and send messages about them to our brain, and control involuntary processes such as heartbeat, the release of hormones, the opening of the pupil in response to light. Neurons send messages by sending electrical signals down the length of its axon, a part of the neuron which we'll discuss later. At the end of the axon, its electrical signal changes into a chemical signal, which is with chemical messengers called neurotransmitters, into the synapse. Neurotransmitters send messages by traveling between cells and attaching to specific receptors on their target cells. Each neurotransmitter attaches to a different receptor. For example, dopamine molecules attach to dopamine receptors. There are several types of neurotransmitters, and some of them are shown in this picture to the left. For example, dopamine, which is involved in feelings of pleasure, learning, focus, and concentration. Serotonin is an inhibitory transmitter, which means it blocks the chemical messages from being transmitted any further. It is involved in regulation of mood, sleep patterns, anxiety, appetite, and pain. The fourth topic we're going to discuss is parts of the neuron. First is the stoma located over here, which basically surrounds the cell and holds its organelles. The stoma is the cell body of the neuron. It contains genetic information, maintains the neuron's structure, and provides energy. Each structure, each each neuronal soma is wrapped in a layer of satellite glial cells containing a nucleus and specialized organelles. The axon of the neuron, located over here, is a long, tail-like structure that joins the cell body at a specialized junction called the axon hillock. They are insulated with fatty substances called myelin, which helps axons conduct electrical signals. Axons help electrical impulses travel between neurons. The nucleus, located over here, is the central part of the neuron and is in charge of producing energy for the cell's functions. Dendrites branching off the nucleus over here are located at the end and form branch-like extensions from the neuron. Its main function is the reception of stimuli from other neurons. Axon terminals, located at the end of the axon of the neuron, are divided into terminals, as shown over here. These terminals link to other neurons to create a synapse. Neurotransmitters are stored here in small areas called synaptic vessels. The transmission of these vesicles from one neuron to other are known as the synapses. Fifth topic, we're going to discuss about the parts of the brain. The brain weighs about 3 pounds in the average adult and is made up of about 60% fat and 40% of a combination of water, protein, carbohydrates, and salts. Before we discuss about the different parts of the brain, we will introduce the topic of gray and white matter in the central nervous system. Gray and white matter are two different regions located in the CNS. In the brain, gray matter refers to the dark outer portion and white matter refers to the lighter inner section underneath. However, in the spinal cord, this order is reversed. Gray matter is mainly composed of somas, while white matter is mainly composed of axons, which are parts of the neuron we discussed earlier. Gray matter is responsible for processing and interpreting information, while white matter transmits information to other parts of the nervous system. Our brains are so complex and consist of several different parts, but through this lecture, we will spend time discussing only the main regions of the brain and their functions to save time. However, feel free to expand your knowledge by researching about the various other regions in your own time. The brain weighs about 3 pounds in average adult, is about 60% fat and 40% of a combination of water, protein, carbohydrates, and salts. At the most basic level, the brain can be divided into three parts: the cerebellum, the brainstem, and the cerebrum. The cerebrum is composed of gray and white matter at its center, and initiates movement and regulates temperatures. Additionally, it is involved in speech, judgment, thinking and reasoning, problem solving, emotions, and learning. The brains the brainstem connects the cerebellum with the spinal cord and includes other regions like the midbrain and the pons. The cerebellum is located in the back of the head and is split into two hemispheres. Its function is to coordinate voluntary muscle movements and maintain posture, balance, and equilibrium. First, we're going to discuss about the parts of the cerebellum. The cerebral cortex is the outer gray matter covering of the cerebellum. It is divided into two hemispheres, the right hemisphere and the left hemisphere. The right hemisphere controls the left side of the body, and the left hemisphere controls the right side of the body. Generally, the left hemisphere controls speech, comprehension, and arithmetic. The right hemisphere controls creativity and spatial ability. These two hemispheres can communicate with each other through the corpus callosum, which is a large C-shaped structure of white matter and nerve pathways located over here. Next, we're going to talk about the parts of the brainstem, which is another lobe of the brain. The brainstem includes the midbrain, pons, and medulla. The midbrain is a complex structure with a range of different neuron clusters and neural pathways. These features facilitate functions such as hearing and calculating responses to environmental change. The, the substantia nigra is located in this area, which is rich in dopamine neurons and affected by Parkinson's disease. The pons is the origin for 4 of the 12 cranial nerves located between the midbrain. This section enables activities such as chewing, blinking, focusing, etc. The medulla, located at the bottom of the brainstem, where the brain meets the spinal cord. This region is essential for survival and regulates bodily activities such as heart rhythm, breathing, blood flow, and oxygen and carbon dioxide levels. The brain is composed of 4 lobes: frontal, parietal, temporal, and occipital lobe. The frontal lobe is the largest lobe and is located in the front of the head. It is mainly involved in personality characteristics, decision-making, and movement. The parietal lobe is located in the middle and is involved with interpreting pain and touch in the body. It additionally helps with identification of objects and spatial relationships. The occipital lobe is located in the back part and is involved with vision. The temporal lobe The temporal lobe is located in the sides of the brain and is involved in short-term memory, speech, and musical rhythm. Other structures within the brain include the pituitary gland, hypothalamus, amygdala, the hippocampus, the pineal gland, and the ventrices. The pituitary gland is a pea-sized structure behind the bridge of the nose. It it owns the flow of other glands in the body, such as the thyroid and adrenal, and regulates the flow of hormones from these glands. The hypothalamus is located above the pituitary gland and sends chemical messages to control the pituitary gland's function. The hypothalamus regulates body temperature, synchronizes sleep patterns, and controls hunger and thirst. The amygdala is a small structure located under each hemisphere of the brain. This structure regulates emotion and memory, stress, and the body's fight or flight response. The hippocampus, located on the underside of each temporal lobe, supports memory, learning, and navigation. This area is affected by Alzheimer's disease, making it harder for someone to form new memories and new information. The pineal gland, located deep in the brain, is involved in responding to light and dark, and secretes the hormone melatonin, which regulates our sleep cycle. The ventrices of the brain manufacture something called cerebrospinal fluid. The CSF is a watery fluid that circulates through the ventrices and spinal cord and, and, and between meninges. This fluid cushions the spinal cord and brain, washes out waste, and delivers nutrients. Sixth topic we're going to discuss is neuron types. First is the unipolar neuron. These neurons have only one cell process extending from the cell body. It has an axon that extends into the dendrites. Unipolar neurons do not exist in humans and only exist in vertebrates such as flies and play a role in the glands and muscles. Pseudounipolar neurons only occur at the sensory neuron. The nerve process extending from the cell body splits into two branches or axons, and therefore the neuron has two separate axons that divide into dendrites at the ends. The bipolar neuron has an axon extending from one side of the body and a dendrite extending from the other. These neurons are uncommon and only occur within a few areas such as the olfactory epithelium and the retina. The most common type of neuron are multipolar neurons, and which are present throughout the CNS. These neurons have many structures extending from their body. There is only one axon, but they have many dendrites branching from the body, making the exchange of information within neurons more efficient. Purkinje neurons also have multiple dendrites that extend from their cell body. They are inhibitory neurons, which means they release neurotransmitters that keep other neurons from firing. Sensory neurons help with experiencing senses such as taste, smell, hearing, vision, and touch. They are triggered by physical and chemical inputs from the environment. Motor neurons help facilitate involuntary and voluntary movement. They allow the brain and spinal cord to communicate with muscles, organs, and glands. Mm, Motor neurons are separated into two types: lower and upper. Lower neurons carry signals from the spinal cord to smooth and skeletal muscles. Upper neurons carry signals between the brain and spinal cord. Interneurons are the most common type of neuron, found in the brain and spinal cord. They pass signals to each other from sensory organs to motor neurons. These neurons form complex circuits that help one react to external stimuli and can also send signals to the pain center in the brain to help experience pain. Next topic is about action potential and synapses. Neurons communicate with each other at junctions called synapses. At the synapse, the presynaptic neuron fires an action potential, which causes the transmission of a signal to the postsynaptic neuron receiving it. Synaptic transmission can be electric or chemical. However, chemical transmission is more common. Chemical transmission involves the release of neurotransmitters, which are chemical messengers that carry information from the presynaptic neuron to the postsynaptic neuron. In the active terminal of the sending cell, synaptic vessels are located. These are membrane bound spheres filled with neurotransmitter molecules. The gap between the axon terminal of the presynaptic neuron and the membrane of a postsynaptic cell is called the synaptic cleft, as seen here. Action potentials are electrical impulses that send signals through the body. To understand how action potentials work, we must first understand how concentration gradients work. A concentration gradient is the difference in ion concentrations, which are charged particles, between the inside and outside of the neuron. At the resting membrane potential, neurons have a negative concentration gradient, which means there are more positively charged ions outside the cell than on the inside. During resting membrane potential, there are more sodium ions outside the cell and more potassium ions inside the cell. However, this concentration of ions isn't static, as ions are flowing in and out of the neuron constantly in order to equalize their concentrations. Now we will discuss the process of action potentials. First is the resting state. During the resting state, which is before the action potential occurs, all the gated sodium and potassium channels are closed. These channels only open once an action potential has been triggered. These channels are called voltage gated, since they are open or closed depending on the voltage differences of the cell membrane. The first main event is when is called depolarization. It is when a triggering signal depolarizes the cell body. This signal comes from other cells connected to the neuron and causes voltage-gated sodium channels to activate, allowing positively charged sodium ions to flow into the negatively charged axon to depolarize it. Depolarizing means to lose the difference of charge between the inside and outside of the cell body. When these channels are activated and sodium ions are sent, the neuron is not at equilibrium anymore and gets more positive, allowing the action potential to pass through. The next stage is called repolarization. Repolarization brings the cell back to its resting membrane potential. The sodium channels close and stop the inward flow of positive sodium ions into the cell. Then potassium channels open, and since there's more potassium inside the cell than outside, more potassium exits the cell. This causes the cell to lose positively charged ions and return back to its resting state. Hyperpolarization occurs when the cell is more negative than its usual resting membrane potential. As the action potential passes through, potassium channels still stay open. Next is hyperpolarization. During hyperpolarization, the cell is more negative than its usual resting membrane potential. As the action potential passes through, potassium channels still stay open, and positive ions continue to exit the neuron. This causes the cell to get more negative than its resting state. Neurotransmitters play a large role in the sending of action potentials. They can be classified as either inhibitory or excitatory. Inhibitory transmitters are sent from the presynaptic neuron and bind to the postsynaptic receptor, but do not generate an action potential to fire the neuron. An inhibitory neurotransmitter essentially prevents the action potential from firing through the other neuron. These neurotransmitters are important in balancing the brain stimulation. An excitatory neurotransmitter does exactly the opposite and promotes the action potential to fire the neuron. These neurotransmitters cause the neuron to become excitable and stimulate the brain. Lastly, we're going to discuss about some of the research methods involved in the field of neuroscience. First is brain lesion studies. This research method involves the examination of patients with brain damage in order to determine which brain structures are damaged and how it influences the patient's behavior. Through this examination, researchers can correlate a specific brain area to an observed behavior through their observations. Next is position emission tomography. This is a method where a nuclear medicine procedure measures the metabolic activity of cells and body tissues. When used in neuroscience, this method can provide 2 and 3 dimensional pictures of brain activity by measuring radioactive isotopes injected into the bloodstream. Next is functional magnetic resonance imaging, also called fMRI. This is a specialized form of MRI and an imaging used to measure the brain's functional anatomy. This method can detect abnormalities of the brain, which cannot be accomplished with other imaging techniques. That was the end of our lecture. We advise you to take notes as the lecture is progressing in order to remember the content. Stay updated to join us for our next lecture on clinical neuroscience.